Hello friends, in today's video I am going to discuss about the important enzyme of glycolysis. For more biochemistry videos, kindly like my channel, that is Tarundev's basic biochemistry classes. So starting with the main part of the video, that is enzymes involved in the irreversible steps of glycolysis. They are hexokinase or glucokinase, phosphofructokinase and pyruvate kinase. This is very important question and it is asked in every competitive exam. So one should know about this, at least these three enzymes of the glycolysis. Now, hexokinase can phosphorylate the hexose, so it can be glucose, fructose, or mannose. Whereas glucokinase specifically phosphorylate the glucose. So you can see alpha D glucose. What is alpha D glucose? When the at the primary carbon hydroxyl group is toward lower side, that is alpha D glucose. This hexokinase or glucokinase will add the phosphate at the sixth position. Now it will be alpha D glucose 6 phosphate. The moment the glucose is converted to alpha D glucose 6 phosphate, it is unable to go out this from the cell. Now in next slide we are going to study about the difference between hexokinase and glucokinase, but major difference between the two is that hexokinase is inhibited by glucose 6 phosphate allosterically. Allosteric is the site other than the active site on the enzyme, and you can see there is a feedback inhibition. So the product form is inhibiting this enzyme which has helped it to synthesize it. So there is a feedback inhibition. Now there are some differences between hexokinase and glucokinase. These are very important. Everyone should know. As I have told you that it is present in hexokinase is present in almost all tissues, whereas glucokinase is present in liver and the beta cell of pancreas. The Km of the hexokinase is very low, so it is having very high affinity for glucose. So it acts on the glucose even at the lower concentration. Vmax is low. In contrast to it, glucokinase is having the high Km and Vmax is high. So it will act when the glucose concentration is more than the 110 or when the glucose concentration is more. So and it is inhibited by glucose 6 phosphate. We have seen because hexokinase is inhibited by the feedback inhibition, whereas no feedback inhibition is seen in glucokinase. And hexokinase is not inducible, whereas glucokinase is inducible by the insulin. Whenever there is a increase in the concentration of the glucose, insulin gets stimulated and insulin will increase the activity of glucokinase. The clinical significance for it is that its deficiency can cause hemolytic anemia, whereas deficiency of glucokinase lead to the diabetes mellitus, which is showing the less activity of glucokinase. Biological significance: hexokinase is involved in maintaining intracellular glucose concentration, whereas glucokinase helps in the blood glucose concentration. So next enzyme is phosphofructokinase. Phosphofructokinase phosphorylates the fructose 6 phosphate with the help of ATP and the magnesium. And you can see this is the sixth position of the fructose. Now the phosphate will be added at the first position of the fructose. Now fructose 1,6 bisphosphate is formed. This word is used when the phosphate is used at the different positions. Now, so here you can see I have mentioned the phosphofructokinase 1 because there is phosphofructokinase 2 also. The function of phosphofructokinase 2 is to convert fructose 6 phosphate to fructose 2,6 bisphosphate. Means the fructose 1,6 bisphosphate is having the phosphate at 1 and 6 position, whereas fructose 2,6 bisphosphate is having the phosphate at the second and 6th position. So when the fructose 2,6 bisphosphate is more, it will stimulate this enzyme. So these are the activators of the phosphofructokinase 1, which include fructose 2,6 bisphosphate and AMP. And inhibitor of phosphofructokinase 1 is ATP, citrate, and phosphoenol pyruvate. We know citrate is intermediate in the TCA cycle. here you can see these are the tandem enzyme. What is tandem enzyme? Tandem enzyme is having the two enzymes which are having the opposite function, but they are formed from the same polypeptide chain. So you can see this is the phosphofructokinase 2 and this is the fructose bisphosphatase 2. So whenever this is dephosphorylated form, in dephosphorylated form, this enzyme, this part is active, that is phosphofructokinase 2 part is active, so it will be forming fructose 6 phosphate to fructose 2,6 bisphosphate. Now glucagon act through the cyclic AMP, so this cyclic AMP will phosphorylate this enzyme with the help of enzyme cyclic AMP dependent protein kinase. So this is ATP is used and it gets phosphorylated. Now we know glucagon play its role when the glucose level is low. So it will activate this fructose bisphosphatase, which will convert fructose 2,6 bisphosphate to fructose, which will stimulate the gluconeogenesis. Gluconeogenesis is the process where glucose is formed from the non-carbohydrate compound, and it will inhibit the glycolysis. But insulin inhibit the function of cyclic AMP, so it will dephosphorylate. Again, this enzyme will get dephosphorylated. So here enzyme is phosphoprotein phosphatase, which help in the dephosphorylation. So now again this phosphofructokinase will be active. So phosphofructokinase 2 will be active, which will be converting fructose 6 phosphate to fructose 2,6 bisphosphate, which will be stimulating the phosphofructokinase. Okay, this stimulates glycolysis and inhibit the gluconeogenesis. Next enzyme is pyruvate kinase. We know that pyruvate kinase, last but not the least, so this is the last step of the glycolysis, and it is the last enzyme which is involved in the irreversible step glycolysis. So pyruvate kinase requires the magnesium and it converts phosphoenolpyruvate to pyruvate, and in this process ATP is formed. The inhibitor of pyruvate kinase, you have to remember 3A, that is ATP, acetyl-CoA and alanine. These are the three main inhibitors of the pyruvate kinase. That's all about the important enzyme or the enzyme involved in the irreversible step of glycolysis. For more videos, kindly like and subscribe my channel, that is Tarun Dev's Basic Biology Classes. Thank you.